So now that we know the introduction and the background for the paper, this next video is going to cover the setup and the study design. There's a few concepts that we have to discuss before we get into what they actually did, and some of these things I'll kind of get at them along the way as we discuss them too. So first I want to talk briefly about this idea of response variables versus treatment variables. Sometimes people will refer to these as the dependent and independent variables. I don't particularly like that terminology because people have a hard time keeping them straight. So I'm going to try to use these terms treatment and response variables. So in an experiment, you impose some kind of treatment or manipulation, and then you see what happens and you measure the outcome. And what you measure in the outcome is the response variable. So I'm going to talk to you about the treatments and the treatment that they set up, and then the responses or the things that they measure. In the last video, I also mentioned something about a natural experiment versus a manipulative experiment. And I want you to think about this paper as having sort of both of these aspects to it. They do a manipulative experiment. They manipulate some things. They set up the number of fish and they put them in these little exclosures. So that's the manipulation, and they actually do an experiment. But then they also take advantage of these naturally invaded ponds, and that's kind of like a natural experiment. No one went out intentionally and invaded these ponds with these fish, or maybe they did, but I don't know that they did. But they're going to take advantage of the fact that those are there, and then they will compare their results of their little manipulative experiment, which is short term, to what they can observe in these natural or more natural settings. And the advantages are that in a manipulative experiment, you can control all the variables and you can run it for a certain period of time. You can really keep track of things. You've got a lot of control, but it's not necessarily representative of what happens in the real world. On the other hand, these observations of real world or big populations, that's realistic, but you give up a lot of control. You don't have any control over how many fish were introduced or how long it's been going on or anything like that. So there's sort of trade-offs of both of those approaches. And part of what they want to do in this paper is they want to compare the results of these two situations. Finally, or the next thing is that they focus on basically a model species. Are they really interested in the species that they're studying in this paper, or are they more interested in expanding and extrapolating these results to all fish in all fisheries? I would argue that they don't care so much about the particular species that they're featuring here, but instead they want to generalize. They want to talk about all sorts of different kinds of fisheries experiments, fisheries settings. And so what they've got is they've chosen 3 model species to work with, and they're sort of hoping that they can expand those results to, or extrapolate those results to other systems. It would be like if we're going to study how a vaccine works on mice as a model organism, but what we're really interested in is not whether it helps the mice, but is this something that could help people. So next, I want to talk about a couple things, more specific methods to this study design in this paper: a substitutive versus an additive experiment. The substitutive experiment is where you substitute one number of individuals for another kind of individual, and we'll talk about this more in a little bit. And an additive experiment is where you just add more and more. You increase the strength of competition by adding more and more individuals. So that's an important concept that we have to cover. And then finally, and this is sort of the biggest thing that blows your mind in this study, is this issue of isotopic niche analysis. And I'm going to get into that a little bit more, especially when we get to the later parts of these slides and then into the results. So first, I think it's always useful to think about what species are involved here. the top one is sort of the focal species. It's the one that they're most interested in. It's a game fish in Europe. It's not a game fish, in other words, a fish that people go after to consume and for sport. It's not a fish that we have here. Goldfish, you've probably all seen goldfish in fish tanks, but goldfish can get to be really, really big fish. This is a relatively small one, so if you do a search for this particular species, you'll find on the internet there's some pretty big ones out there. So goldfish can be really big, and I'm gonna call these by their common names from here on out. So we've got tench, the sort of focal or species of interest, and then the manipulated species, goldfish and carp. Carp is a species that we have in our waters here. There's a few different species of carp, but we have these throughout North America, and they also can get quite large. So 3 species that have similar feeding requirements. As the paper says, they are plastic in their diet, which means they're somewhat flexible in their diets. And so the authors selected these species as ones that they thought would, be the most likely to show responses that they're interested in seeing. If any fish could respond in the way that they expect, it would be these. So then they also talk about their experimental setup. They've got these exclosures, or enclosures I should say. Exclosures keep things from coming in. Enclosures keep things from leaving. So in the lower picture here, we've got a field researcher who's studying plants or fish that are on the inside of that enclosure. So they've got some ponds, and they set up multiple of these enclosures, and they stock them with different concentrations of fish. And the idea here is it's semi-natural, because you've got these fish that are out in the lake, and natural food is kind of moving in and out through these grates, and there's plants that are coming in and insects that are coming in, and the fish that are inside of those enclosures are restricted and they can't go anywhere. So you can find them and measure them again later on. And then ultimately they're going to compare those results to what happened in natural ponds. Now I wasn't able to find a whole lot of information on these natural ponds, but these are pictures of what I would imagine are ponds, and they're from that same region. So these ponds probably contain at least some of these species of fish. And again, the idea is they're going to compare the results of what they see in these small enclosures to what they see in these larger ponds, and that's sort of their third point of interest. And you could think of this as maybe a natural experiment, although it's really just observational data that they take from some situation that's out in nature. So in their text on page 488, under the materials and methods, they talk quite a bit in that first paragraph about the setup that they highlight in this table. And so I want you to try to follow along. This is section 2.1 on page 488, really that first paragraph here. They talk a lot about their substitutive versus additive treatments. So here's the table with some annotations on the side. The top 3 conditions are each species by itself. And so allopatry means when they're living by themselves. The top species, T. tinca, is the tench. The next row is the carp, and then the bottom one, or the third row, is the goldfish. And so these are the reference conditions that they're going to compare these other things to. Their manipulated treatments involve these things called substitutive treatments. Here we've got 4 individuals of tench and 4 individuals of carp. 4 tench, 4 carp, for a total of 8. So what they've done is they've essentially removed 4 of the individuals here and replaced them with 4 individuals of a competitor. Then they did the same thing with by removing or retaining, we'll say, 4 individuals of tench, now 4 individuals of goldfish, for a grand total of 8 individuals. Same thing here, so now we've got carp and goldfish, and the total number is the same as above, with the idea being um, we're not necessarily increasing the strength of total consumption here, but we're just changing the identity of the species who are doing the competing. So these would be sympatric in that there's two species together, and these are also obviously interspecific treatments. They refer to this one last in their paragraph in section 2.1, so it's odd that they put it here in the table. Nonetheless, here it is. So we'll come back to that because they refer to it last. The other set of treatments are the additive treatments. So here we've got interspecific competition, just like up here. But here we've got 4 of each species. So it's kind of like sympatry, except we've got an additional 4 individuals of the other species. And here we've got interspecific competition. Again, it's all 3 species, but it's doubled from before. And here we've got interspecific competition. In the strongest case, they are really stocking a lot of individuals in there. And so we've got a total of 36 individuals. So these are additive treatments in that they're adding more individuals. These are substitutive treatments in that they're sort of replacing some individuals with individuals of another species. Finally, this final treatment here is where they have very strong intraspecific competition. So they've basically taken tench by itself and then added more tench individuals. So that's kind of like another reference treatment. So the next thing in terms of what we need to get our heads around for the setup of the experiment is what are the responses. So they've got their treatment variables. It's the levels of competition and the identities of the species, but then they've got the things that they measure at the end. What are the outcomes of this experiment? And so they measure a lot of stuff, but really try to think of it as just a couple things. First of all, what did they measure? They measured final fish weight. All the fish started out at about the same weight, and so they are just calculating the final, they measure the final mass. That allows them to calculate growth rate. How big did these fish get during a certain period of time? Now fish have this thing that's called indeterminate growth. In other words, they keep growing until they die. They get bigger and bigger and bigger until they die, unlike humans and dogs and things like that. So they will keep growing. So you can just measure their final growth rate, and that gives you, or final weight, and that gives you the size of the total fish and how much they grew. They also measured these things called isotopic ratios. Now let's go down to here. So they measure nitrogen normally has number of protons as 14, but there are some isotopes of nitrogen in the atmosphere, a very, very small percentage, which have 15 protons. And different kinds of organisms have sort of different ratios of those N14 versus N15 isotopes. Plants will have a different ratio than will consumers or herbivores. Predators have a different ratio than either plants or herbivores. So the nitrogen 15 isotope ratio is something that allows you to determine like what has that organism been eating. They give us a reference condition for nitrogen where 2, a measurement of 2 of an N15 ratio, indicates that these things are basically macroinvertebrates. So these insects, macroinvertebrates, have an isotopic ratio of about 2. That's kind of a reference that we'll use later on. And then carbon is the same kind of situation. Carbon normally has 12 protons, 12 neutrons, but there are some tiny fraction of carbon out there that exists with 13 neutrons, and that is C13. C13 is discriminated against at different levels or different strengths in plants versus herbivores versus predators. So it also is something that's useful for determining something about um, how the organism feeds. According to something that they've kind of hidden in the results, more negative values, say like -30, means that a species is more predaceous. Larger values, in other words, less negative, like -24, tend to be more herbivorous. We may or may not get into those details too much later on. I don't know that it's super important, plus they're also not super clear on it. But that is just useful as a reference. If we go back up here, so they've used these isotopic ratios as some kind of index of trophic position. So are you eating more other insects or are you eating more plants? And trophic size or niche size. And I need to define this idea of index here. Sometimes you measure very directly the thing that you're interested in. So imagine that we were to put you on a particular diet where you only get to eat 1,000 calories a day. And what would happen is you would lose weight. Well, how do we measure that? Well, you could weigh somebody. That's a direct measure of how much weight they're losing. But you could also take an indirect measure or an index of how much weight they're losing. If you lost a whole bunch of weight, you can imagine that your face would look thinner. That's an index of you losing weight. You can imagine that your clothes don't fit you as well. That's an index of you losing weight. So it's an index is not a direct measure. Instead, it's sort of an indirect measure of the thing you're actually interested in. So given that, the SEA, the standard ellipse area, is kind of a combination of the nitrogen 15 ratio and the carbon 13 ratio that is an index of the population's typical resource use. In other words, it is a measure or an index of their niche. And remember that niches are multi-dimensional things, so this niche has two particular dimensions, the nitrogen 15 dimension and the carbon 13 dimension. And we'll talk about those a little bit more in the next video when we get into results.